Hi students, welcome back to GDC classes. So in the last session, we stopped while discussing the stomach. Okay, we stopped while discussing the stomach, and in this session, let us start with the stomach itself. I have told you that the stomach, the inner lining, that is the mucosal lining of the stomach, contain different types of cells, the exocrine cells and also the endocrine cells. The exocrine cells will secrete different fluids and enzymes into the stomach, whereas the endocrine cells will secrete different hormones into the stomach. So let us know about the cells. So the exocrine cells present in the mucosal lining of the stomach or the mucus cells, chief cells, and the parietal cells. Whereas the endocrine cells or the G cells, D cells, and enterochromaffin like cells. So coming to the chief cells, each type of cell will secrete different types of enzymes and different types of fluids. So what does the chief cells do? The chief cells will secrete pepsinogen, renin, and lipase. So these are the three types of enzymes which the chief cells will secrete into the stomach. Now the parietal cells will secrete the hydrochloric acid and intrinsic factor. And mucus cells, they will ultimately secrete the mucus. G cells. Coming to the endocrine cells, the G cells will secrete the gastrin hormone, D cells will secrete the somatostatin hormone, and the enterochromaffin like cells will secrete the histamine hormone. So remember what cells each certain type of cell will secrete certain type of enzymes and hormones. Now coming to the mucus cells, let us discuss in detail. Mucus is produced by the mucous neck cells and also the surface mucus cells. So here I will show you. So I have told you that the inner lining of the stomach is present in the form of folds. So these are the folds. So these are the folds. So if we zoom into a single fold, the surface, this is the surface, right? This is the surface. The surface has surface mucus cells and the neck portion has the mucous neck cells. So these two types of cells, that is the surface mucus cells and the mucosal neck cells, they will secrete the mucus. And the HCl, that is the hydrochloric acid, which is a highly acidic substance, it is secreted in the stomach. Now the mucus secreted by these mucosal cells will form a protective lining to the inner wall of the stomach, preventing any damaging or corrosive effects of the HCl on the inner lining of the stomach. Okay, this mucus will protect the inner wall of the stomach from the corrosive actions of the hydrochloric acid. So that is the function of the mucus. Now coming to the hydrochloric acid, it is secreted by the parietal cells of the stomach. It destroys the harmful bacteria. So when we eat food, along with the food, we might also ingest certain bacteria or certain germs. So this hydrochloric acid will, as it is a highly acidic substance, it will help in killing the bacteria, and it also activates inactive enzymes. Okay, inactive enzymes means these are the enzymes which won't be able to function until unless they are converted into their active forms. So, for example, pepsinogen. So, pepsinogen is an enzyme that is secreted by the chief cells of the stomach. Now, this pepsin is present. Pepsinogen is an inactive enzyme. This pepsinogen, in the presence of HCl, gets converted to its active form, that is pepsin. Now, this pepsin is a protein digesting enzyme which will digest the proteins or break down the proteins into small peptide chains, that is dipeptides, tripeptides, and these peptides will be further broken down into amino acids. Okay, HCl, the function of the HCl is to activate the inactive enzymes into active enzymes. Now coming to the other substance that is secreted by the parietal cells of the stomach is the intrinsic factor. Now, this intrinsic factor is a glycoprotein secreted by the parietal cells. It is necessary for the absorption of the vitamin B12. So, this intrinsic factor is necessary for the absorption of vitamin B12. Now, vitamin B12 is necessary for different functions, like for the formation of the DNA and for maintaining the nerves and the different cells of the body in a healthy condition. The vitamin B12 is required. And if the vitamin B12, if it has to be absorbed into the blood circulation, the intrinsic factors presence is a must. So the vitamin B12, we get the vitamin B12 from the food we eat. And the intrinsic factor is secreted by the parietal cells of the stomach. And these two will form a complex called the intrinsic vitamin B12 complex. And this complex is absorbed in the small intestine. The complex is formed in the stomach, but it is absorbed only in the small intestine. Vitamin B12 will be absorbed only in the presence of intrinsic factor. If the intrinsic factor is not secreted properly by the parietal cells, then the vitamin B12 deficiency will occur in the body, which can lead to different other conditions in the body. Sorry. Now the next enzyme is the gastric lipase. It is secreted by the chief cells of the stomach. So it is a lipid digesting enzyme or fat digesting enzyme. You know that the fats or the lipids are present in the form of triglycerides. Triglyceride means it contains three fatty acids attached to a single glycerol moiety. So to this glycerol moiety, three fatty acid chains are attached. Lipids occur in the form of triglycerides. The triglycerides in the form of lipases, they will be broken down into diglycerides and fatty acids. And the diglycerides are further broken down into monoglycerides and fatty acids. So fatty acids are the simplest forms of lipids. And these fatty acids can be easily absorbed into the lymphatic circulation. And remember one thing, majority of the fats are digested and absorbed only in the small intestine. That is about the gastric lipase. Now coming to the renin. Renin is a protein digesting enzyme which is secreted by the chief cells of the stomach. Okay, and it is secreted in its inactive form called prorenin. So prorenin, it is secreted by the chief cells in the form of prorenin. Prorenin is an inactive enzyme, and in the presence of hydrochloric acid, it is converted to active form called renin. What is the function of this renin? It converts the milk protein, that is the casein protein, into insoluble uh, casein. Okay, it will help in the digestion of milk protein, that is casein. Okay, prorenin in the presence of HCl gets converted to renin, which is the active form, and the renin will try to digest the casein protein, which is particularly present in the milk, into peptones and proteases, which are simpler forms of casein protein. And now coming to the hormones. So until now we have discussed about the enzymes and different fluids, that is the hydrochloric acid, intrinsic factor, prorenin, pepsinogen, and the lipase. So these are the things or the fluids secreted by the exocrine cells of the stomach. Now, the endocrine cells of the stomach secrete different hormones. Let us know about them. So, the gastrin is the peptide hormone secreted by the G cells present in the stomach inner wall. And the gastrin hormone binds to the cholecystokinin B receptor on the surface of the parietal cells and stimulate the release of HCl. So, these G cells, which are present in the gut wall, they will release the gastrin hormone into the bloodstream, and this gastrin hormone will again re-enter into the gut wall, and they will bind to the gastrin hormone will bind to the cholecystokinin B receptor present on the surface of the parietal cell, and which will ultimately cause the release of hydrochloric acid. So that is the function of the gastrin. It will cause the release of hydrochloric acid from the parietal cells. Now the other hormone is the histamine. It is released from the enterochromaffin-like cells present in the gut wall. And what is the function of the histamine? It will also cause the parietal cells to release the hydrochloric acid. So the G cells present in the gut wall will release the gastrin hormone. The gastrin hormone will stimulate the enterochromaffin-like cells to release the histamine hormone, and the histamine hormone will bind to the H2 receptor present on the parietal cell, surface of the parietal cell, and the parietal cell will in turn release the hydrochloric acid. So this is the function of the histamine. Now coming to the somatostatin. So somatostatin is a hormone released by the D cells. And what is the function of this somatostatin? It will prevent or it will cause a decrease in the release of the hydrochloric acid. So what it will do? It will prevent the parietal cells from releasing the hydrochloric acid. It will prevent the enterochromaffin cells from releasing the histamine hormone. It will prevent the G cells from releasing the gastrin hormone. So, as a result, the gastric acid secretion will also be decreased. So, conclusively, you can say that the gastrin hormone and the histamine hormone will cause an increase in the HCl production, whereas the somatostatin hormone will cause a decrease in the production of the hydrochloric acid. So that is about the different enzymes and hormones secreted by the cells of the stomach. Now coming to the diseases associated with the stomach. First one is the peptic ulcer. It is an infection in the wall of the stomach or the duodenum, and it is caused commonly by a bacteria called Helicobacter pylori, or it may be also caused by the drugs like the anti-inflammatory drugs like aspirin. So the peptic ulcer, it is an infection to the wall of the stomach. It can be caused due to a bacteria called Helicobacter pylori, or it can also be caused due to the prolonged intake of anti-inflammatory drugs like aspirin. So, what these anti-inflammatory drugs do, they will decrease the production of the mucus secretion. Okay, they will decrease the production of mucus. What the mucus will do? The mucus will protect the inner wall of the stomach from the corrosive actions of the HCl. And when the production of the mucus is decreased, the protective mucus lining will not be formed, and as a result, the inner wall of the stomach will be exposed to the HCl, causing the peptic ulcers to happen. And coming to other disorder, that is gastritis. Gastritis means it is the inflammation of the lining of the gut, and it is caused due to smoking, excessive drinking of alcohol, or any chronic infection, or long-term usage of drugs and medicines. So it is the inflammation of the inner lining of the stomach, which can be caused due to smoking, chronic alcohol intake, and also chronic intake of drugs and medicine. Now coming to, let us discuss the accessory digestive organs, that is the pancreas. Pancreas is about 12 to 15 centimeters long, and it is a soft elongated organ situated between the limbs of the duodenum. So here between the limbs the pancreas is present. It is an elongated organ which is divided into three portions: the head portion, the body portion, and the tail portion. And the pancreas is a heterocrine gland. So, what does it mean? It performs both endocrine functions and exocrine functions. Exocrine part of the pancreas will secrete pancreatic juices and pancreatic enzymes into the small intestine, while the endocrine portion of the pancreas will cause the production or release of hormones into the blood circulation. So, what does the exocrine part of the pancreas do? It will cause the secretion of pancreatic juice into the small intestine, and endocrine portion will cause the secretion of hormones like insulin and glucagon into the blood circulation, which will influence the levels of glucose in the blood. But here in this session, we will concentrate on the exocrine part of the pancreas. 95% of the pancreas consists of exocrine portion. Okay, the remaining 5% will constitute the endocrine portion. And what does the exocrine portion constitute? It contains small clusters of cells called acini. Okay, these cluster contains small cells called acinor cells. Okay, I'll show you. So these clusters here, this is the acini, and the acini contains a number of cells called acinor cells. In these acinor cells, all the pancreatic juices, pancreatic enzymes will be synthesized, and from these acinor cells, the secretions will pass or drain into the pancreatic duct. So here, this passage is known as the pancreatic duct. All the secretions of the pancreas will drain into the pancreatic duct. And from the liver, right, the liver will synthesize the bile juice, and the bile juice will be stored in the gallbladder. Okay, the bile juice will be stored in the gallbladder, and from the gallbladder, it will enter the cystic duct and then into the common bile duct. The common bile duct combines with the pancreatic duct and forms the hepatopancreatic ampulla, or you can also call it as ampulla of water. Okay, the common bile duct and the pancreatic duct will combine to form the hepatopancreatic ampulla or the ampulla of water. And this ampulla of water will release the secretions of the pancreas and the gallbladder into the small intestine. And this ampulla of water is guarded by a sphincter called sphincter of Oddi. So remember these names, okay? You have to remember the names. The ampulla of water will release the secretions of the pancreas and the bile juice into the small intestine. And this ampulla of water is guarded by a sphincter called the sphincter of Oddi. So here in this, yeah, okay, I'll show you. So this dilated vessel, which will cause the release of the secretions into the small intestine, is known as the ampulla of water, which is formed by the combination of common bile duct and the pancreatic duct, and it is guarded by a sphincter called sphincter of Oddi. And now let us know the composition of the pancreatic juice. The pancreatic juice is a clear, colorless fluid with a pH between 7.1 to 8.2. So you can say this as a basic pH. And about 1200 to 500 ml of pancreatic juice or pancreatic secretion is produced per day into the small intestine. What does the pancreatic juice contain? It contains water, mineral salts, sodium bicarbonate, enzymes like amylase, lipase, and inactive enzymes like trypsinogen, chymotrypsinogen, and procarboxypeptidase. So this pancreatic juice is a highly alkaline fluid. Why it is alkaline? It is because of the presence of sodium bicarbonate, which is a basic compound. Now, the trypsinogen is an inactive enzyme. Trypsinogen, in the presence of an enzyme called enterokinase, so when the pancreatic juice, when it is secreted into the small intestine, the cells of the small intestine secrete an enzyme called enterokinase. This enterokinase enzyme will convert the trypsinogen, which is an inactive enzyme, into trypsin. And this trypsin in turn will activate the chymotrypsinogen into chymotrypsin and procarboxypeptidase into carboxypeptidase. Okay, that is about the pancreatic juice. Now let us discuss the disorders of the pancreas. Pancreatitis is the swelling or the inflammation of the pancreas. It may be chronic or acute, and it is caused due to heavy alcohol intake or gallstones. So gallstones are nothing but they are they will cause interference or they will cause some obstruction in the ducts. So here these are the gallstones. They will cause some interference to the flow of bile from the gallbladder. So bile is stored in the gallbladder. So when the gallstones are present in the ducts, causing some interference or obstructions to the flow of the bile, all the bile synthesized from the liver will get accumulated in the gallbladder, causing the pancreas and also the gallbladder to swell, to inflame. Okay, when there is presence of gallstones, the pancreatic juices and also the bile juices, they will not flow into the small intestine properly, and as a result all the pancreatic juices and also the bile juices will get accumulated in the pancreas and the gallbladder, and as a result the pressure will build up and resulting in the inflammation of the pancreas. Now coming to a condition called steatorrhea. Steatorrhea or is also known as fatty stool. So if the pancreas is not working properly, the pancreas won't be able to release an enzyme called pancreatic lipase. Now, this pancreatic lipase is an enzyme which will help in the digestion of lipids. And if the pancreatic lipase is not released properly, the lipids present in the food will not be digested and they will not be absorbed. And as a result, what happens? All the lipids present in the food will be excreted out in the form of feces, resulting in a oily appearance of the feces. So if the feces appear oily, that means that the pancreas are not functioning properly or the pancreas are not properly releasing the pancreatic lipase enzyme. So one thing I've, I forgot to mention, the pancreas has an additional duct called the accessory pancreatic duct. So when the pancreatic juice is secreted in the pancreas, it enters into the pancreatic duct. All the pancreatic secretions will enter into the pancreatic duct and will build up pressure in the pancreatic duct. So if all the secretions of the pancreas enter only into the pancreatic duct, pressure will build up in the pancreatic duct. So in order to decrease this pressure buildup, an accessory pancreatic duct is present, which will cause the decrease in the pancreas. So some part of the pancreatic juice will also travel into this accessory pancreatic duct, resulting a decrease in the pressure buildup in the pancreatic duct. So that is about the pancreas. Now coming to the liver. Liver is the second largest organ. So what is the first largest organ in the body? It is the skin. Now the liver is it is around 1.4 kgs in weight, and it is located upper right hand portion of the abdominal cavity beneath the diaphragm and on top of the stomach. So it is present on top of the stomach and it is present on the right side of the body. It is divided into two lobes. So a ligament called, so this is the ligament, a ligament called falciparum ligament will divide the liver into two lobes, that is the right large lobe and the left small lobe. The liver has a number of cells called hepatocytes. Okay, hepatocytes will synthesize the bile, and this bile will enter into the intrahepatic ducts present between the hepatocytes. Okay, all the bile which is synthesized in the hepatocytes will enter into the intrahepatic ducts, and from the intrahepatic ducts they will enter into the right hepatic duct and left hepatic duct. So this is the right hepatic duct, this is the right hepatic duct, and this is the left hepatic duct. And these right and left hepatic ducts, they will combine and drain into the common hepatic duct from the liver. Common hepatic duct from the liver. So this is the right hepatic duct, this is the left hepatic duct. They will combine to form the common hepatic duct. This is the common hepatic duct, and the common hepatic duct will combine with the cystic duct from the gallbladder and forms the common bile duct. So this is the cystic duct, this is the common hepatic duct which will combine to form the common bile duct. And this common bile duct combines with the pancreatic duct and forms the ampulla of water which opens into the duodenum. So remember this pathway. The bile synthesized in the liver will enter into the intrahepatic duct. From the intrahepatic duct, they will enter into the right and left hepatic ducts. These will combine to form the common hepatic duct. The common hepatic duct will combine with the cystic duct of the gallbladder and form the common bile duct. And the common bile duct in turn combine with the pancreatic duct and forms the ampulla of water which will open into the duodenum. And the functions of the liver performs a variety of functions which are very important to maintain the body in a normal condition. So it is involved in carbohydrate metabolism. That means it is involved in glycogenolysis, gluconeogenesis, and glycogenesis. What is glycogenolysis? It is the breakdown of the glycogen into glucose molecules and the conversion of these glucose molecules into ATP, that is into energy. So whenever there is a need for the body, a need of energy for the body, the glycogen is broken down into glucose molecules and these glucose molecules are converted into ATP molecules. And gluconeogenesis means it is the synthesis of glucose from sources other than glycogen, like from proteins, from lipids, the glucose will be synthesized. And glycogenesis means it is the synthesis of glycogen. So when there is no need of energy for the body, all the glucose that we get from the food and the excess glucose that is present in the blood circulation, it will be converted to glycogen and it will be stored in the liver cells and also in the muscle cells. And it is also involved in lipid metabolism. Lipid metabolism means it is the breakdown of the fatty acids to generate the ATP molecules. And also the liver helps in the synthesis of lipoproteins. These lipoproteins will help in the transport of fatty acids from the small intestine to the lymphatic circulation. And it is also involved in the protein metabolism, detoxification. Detoxification means it will help in removing the toxins from the blood, from the body. So, for example, if we take a certain medicine, the medicine will perform its pharmacological activity, and after its pharmacological activity, it has to be eliminated out of the body. And who will do that function? The liver will do that function. The liver will help in eliminating the drug after its activity in the body. That is the detoxification. And excretion of bilirubin. So bilirubin is a bile pigment which is formed as a result of a process called hemolysis. Hemolysis means it is the breakdown of the old RBC. So you all know that RBC has a lifespan of about 120 days. And after these 120 days, the RBCs will undergo a process called hemolysis. It is the breakdown of the RBC. And as a result of the RBC hemolysis, the bilirubin is formed as a byproduct. And this bilirubin will be eliminated into the bile and then out of the body. And it also synthesizes bile salts which are required for the digestion and absorption of fats present in the food and also in the storage of glycogen and certain vitamins. It contains certain cells called Kupffer cells, which are a type of immune cells. They will, they will perform the phagocytosis of the old RBC cells and also bacteria. So when bacteria enters our body, these Kupffer cells will engulf those bacteria and kill those bacteria. And also the when the RBCs complete their lifespan, the Kupffer cells will engulf those old RBCs. And then the livers also involved in the synthesis of vitamin D. So along with the skin and the kidney, the liver is also involved in the synthesis of vitamin D. Now let us talk about the gallbladder. Now this gallbladder is a pear-shaped sac whose main function is to act as a reservoir or a storage site for the bile juice. So the bile juice is synthesized in the liver and it is stored in the gallbladder. Now this gallbladder is present posterior to the liver. It is attached to the posterior surface of the liver. Let us know about the bile composition. Bile is a physiological solution produced by the liver, and it is a fluid which contains about 95% of the water, and 5 the remaining 5% constitutes different solid constituents like the bile salts, taurochenodeoxycholic acid, and glycochenodeoxycholic acid. So these are the bile salts present in the bile, and then the bile acids, taurocholic acid, glycocholic acid. And bilirubin, it is the byproduct of broken down RBCs. As I have told you earlier, bilirubin is a byproduct of hemolysis process. And also the bile contains some amounts of cholesterol. So this is the bile composition. Major part of the bile is composed of water, and the remaining portion has bile salts, bile acids, bile pigments, and cholesterol. What is the function of the bile? It will help in digesting the and absorption of fats. The bile salts, particularly, they will help in the digestion and absorption of fats. Excretion, heavy metals, bacteria, bile pigments, and excess cholesterol. These things will be excreted into the bile, and then it will be excreted out of the body. And it also acts as a laxative. So laxative is a substance which will cause or which will stimulate the bowel movement, which will cause excretion of the feces. So the bile will exert some stimulating action on the intestinal walls, resulting in the propulsion of the food or the digested food through the towards the large intestine. Neutralization. Bile, the bile is an alkaline fluid. Okay, when it enters the small intestine, it will try to neutralize the acidic pH of the chyme that enters the small intestine from the stomach. So the stomach, along with the food, the hydrochloric acid also enters the small intestine, and the chyme has an acidic pH. Now, as the bile is an alkaline fluid, it will help in the neutralization of the acidic pH of the chyme in the small intestine. Now let us discuss about the small intestine. It is about 5 meters long. The small intestine is a supercoiled structure which begins at the pyloric sphincter. So the entry of the food from the stomach into the small intestine is guarded by a sphincter called pyloric sphincter. And the entry of the food from the small intestine into the large intestine. Okay, one second. Okay, sorry. The entry of the food from the stomach into the small intestine is guarded by a sphincter called the pyloric sphincter, and it is present in the abdominal cavity surrounded by the large intestine. So this is the small intestine here, and it is surrounded by the large intestine. This is the large intestine. The small intestine is divided into three portions: the duodenum, the jejunum, and the ileum. Duodenum is about 20 to 25 centimeters in length, jejunum is 2 meters in length, and ileum is 3 meters in length. So, from this, you can say that ileum is the longest portion of the small intestine. And the innermost layer, that is the mucosal layer of the small intestine, is present in the form of circular folds. So, here, these are the circular folds, and they extend up to 1 centimeter into the lumen of the small intestine. So they can extend up to 1 centimeter into the lumen of the small intestine. And these folds, they contain, on the surface of these circular folds, they contain several small finger-like structures called villi. Okay, on the folds are covered by small finger-like structures called villi. And these villi, they contain different types of cells like the enterocytes, goblet cells, which are involved in different functions. Like the enterocytes, they are the absorptive cells. That means they are involved in absorbing the nutrients from the small intestine into the blood circulation. And the goblet cells, they will secrete the mucus. Okay, goblet cells are involved in mucus secretion. Enterocytes or the absorptive cells are involved in absorbing the nutrients. And there is a gland called Brunner's gland, which is present in the submucosal layer of the intestinal wall, and it secretes the alkaline mucus. So when the stomach content, that is the chyme, enters the small intestine, it contains the acidic pH, right? So the Brunner's gland will secrete the alkaline mucus, which will neutralize the acidic pH of the chyme. And the intestinal glands present below the surface of the villi. So here, below the surface of the villi, the intestinal glands present are known as crypts of Liberkin. Okay, these crypts of Liberkin, that is the intestinal glands present below the surface of the villi, they will secrete the intestinal juices. And these intestinal juices will contain different enzymes like the peptidases, which is the enterokinase. So if you remember earlier, I have told you that enterokinase is the enzyme secreted in the small intestine, which will activate the trypsinogen enzyme into trypsin. Okay, remember. And along with enterokinase, it also has sucrase, which is a sucrose digesting enzyme. Maltase digests the maltose. Lactase will digest the lactose. Lipase will digest the lipids. Amylase will digest the starch. And aminopeptidase and carboxypeptidase will digest the proteins. Like the stomach, okay, stomach will produce both enzymes and hormones. Okay, like the stomach, the small intestine will also produce enzymes and hormones. And what are the hormones secreted by the small intestine? It is the cholecystokinin and the secretin. So here, the cholecystokinin is the enzyme which will stimulate the gallbladder to contract and thus increase the flow of bile salts into the small intestine. So, in the food we eat, lipids will be present, right? So, the presence of lipids in the food will stimulate the intestinal glands to release the cholecystokinin hormone. And this cholecystokinin will cause the gallbladder to release the bile juice into the small intestine. And these bile juice, okay, bile juice contains bile salts, which will in turn cause the digestion and absorption of fats. So it is like a cycle. And the secretin hormone will stimulate the release of alkaline pancreatic fluid in order to neutralize the stomach acid that enters the small intestine. So the cholecystokinin will cause the release of bile into the small intestine, aiding in the digestion and absorption of fats, whereas the secretin will cause the release of pancreatic juice into the small intestine. Now let us discuss the digestion of lipids. Okay, the fats or the lipids will be present in the form of triglycerides. That is, a single glycerol moiety is attached to three different fatty acids. And these lipids, they will combine with the bile salts and form emulsion droplets. Okay, the lipid molecules will combine with the bile salts and form emulsion droplets. And after these emulsion droplets are formed, the pancreatic lipases will cause the digestion of the triglycerides and form fatty acids. And this process of digesting the triglycerides or breaking down of the triglycerides into fatty acids is known as emulsification. Okay, after emulsification, micelles will be formed. Micelles will be formed. Micelles is a combination of bile salts, monoglycerides, and fatty acids. These micelles will enter into the enterocytes, that is the cells of the small intestine. And in the enterocytes, the micelles will mature into chylomicrons. So chylomicrons are nothing but small, small ball-like structures which contain bile salts, monoglycerides, fatty acids, phospholipids, and cholesterol. So these are the chylomicrons. It is the structure of the chylomicron. And in the enterocytes, once the chylomicrons are formed, they will transport the fatty acids into the lymphatic circulation. So remember one thing, the villi present in the small intestine has both lymphatic supply and blood supply. That means it has both lymphatic capillaries and blood capillaries. The lymphatic capillary is particularly known as lacteal. So this structure, this main vessel is known as lacteal. So the chylomicrons from the cells of the intestine, that is from the enterocytes, it will enter into the lymphatic circulation, that is the lacteal. So this is how the digestion and absorption of fats will takes place in the small intestine. So majority of the fats will be digested only in the small intestine. So in the saliva, salivary lipase will digest some part of the lipids, and in the stomach the gastric lipase will digest the lipids, and in the small intestine the pancreatic lipase will cause the digestion of major amount of the lipids present in the food. And a particular thing I have forgot to told you to tell you is that the bile salts, okay, the bile salts will combine with the lipids and they will form the micelles. And once the bile salts enter the enterocytes, they will not be absorbed into the lymphatic circulation. So the bile salts, they will not be absorbed into the lymphatic circulation. They will remain in the enterocytes, and in the ileum of the small intestine, they will be absorbed into the blood circulation, and from the blood circulation, they will enter into the liver. And the liver, again, the bile salts will again it will enter into the smaller intestine, and again they will be used in the digestion of the fats. So this cycle, it is known as the enterohepatic circulation. So from the enterocytes, from the small intestine, the bile salts will enter the blood circulation. From the blood circulation they will enter the liver, and from the liver through the bile it will again enter into the small intestine. So it is a cycle. Now, coming to the large intestine, the large intestine surrounds the small intestine and it is about 1.5 meters long. So, here you can see the cecum, the colon, the rectum, and then the anus. Okay, the large intestine is divided into four portions. The cecum, the colon is again divided into, you know, ascending colon, transverse colon, and descending colon. So, here you can see ascending, transverse, and descending colon, and then the rectum, and finally the anus. The entry of ileum, that is the entry of the small intestine into the large intestine, is guarded by a sphincter called the 𝙂𝙀𝙎𝑐𝑒𝑐𝑎l sphincter, or you can also call it as Bowhin wall. Okay, the entry of the small intestine, the entry of the stomach, food from the stomach into the small intestine is guarded by a sphincter called pyloric sphincter, and the entry of food from the small intestine into the large intestine is guarded by a sphincter called 𝙂𝙀𝙎𝑐𝑒𝑐𝑎l sphincter, which we can also call as Bowhin wall. And there is a structure called vermiform appendix. It is a muscular worm-like structure attached to the cecum, attached to the cecum of the large intestine. So it is said to be a vestigial organ. Vestigial organ means it doesn't perform any function. Okay, that is about the parts of the large intestine. And what is the function of the large intestine? All the villi, okay, villi are present only in the small intestine, and all the absorption of the nutrients will take place only in the small intestine. And villi are not present in the large intestine, so the absorption of nutrients will not happen in the large intestine. And what will happen in the large intestine is that all the remaining water, remaining electrolytes, it will be absorbed in the large intestine, and it will make the food to solidify. Okay, if all the water and all the electrolytes are absorbed from the food, from the remaining food in the digestive tract, the food will solidify and it becomes somewhat solid. And a large intestine, it accommodates a number of useful bacteria like the E. coli, Streptococcus faecalis, Enterobacter aerogens. So these bacteria, they are useful for the human body and they will help in the synthesis of vitamin K. and this vitamin K is in turn used in the synthesis of blood clotting factors. So vitamin K is useful for blood coagulation, and in the small intestine, as the food becomes solid, as it solidifies, it enters into the rectum. Okay, when the rectum gets full, a nerve signal will be produced. A nerve stimulus will be triggered, asking the contents of the rectum to be emptied into the anal canal. So the solid constituents, that is the waste present in the rectum, when the nerve is triggered, it will enter into the anal canal. Now the anal canal is guarded by an internal smooth muscle sphincter and an external skeletal muscle sphincter. The opening of the internal smooth muscle sphincter is in the control of the nervous system. We cannot control the opening of the internal smooth muscle, whereas the external skeletal muscle sphincter of the anal canal can be controlled. The opening of that sphincter can be controlled by the human. Okay, full, whenever the rectum is full with the waste material, the nerve stimulus will be triggered, and the waste will enter into the anal canal. The internal smooth muscle sphincter will open. The waste will enter from the rectum into the anal canal, and if the external sphincter is opened, the waste will be excreted out of the body. And in the large intestine, some mucus will be secreted, which will help in the easy removal of the waste from the rectum of the large intestine. So this is the function of the large intestine and overall the digestive system. So that is the digestive system. We have discussed everything about the digestive system so far. Thank you.